So welcome everyone to the second year of a hybrid meeting for the 2023-2024 JAM year.
Okay, so this is
I am so excited to see so many smiling, excited faces, to hear all the science there is to hear, and to see that we have plenty of participants online. Thank you for joining. So what we're going to go through is a little bit of announcements, and then get to the presentation, right? So what I want to start off with, though, is something new. I want to start a membership highlight for JAM. So I know that so many of our JAM membership does amazing things day in and day out in the laboratory. Sometimes they don't think that it's amazing because our technologists are very humble and think it's just part of the work, but that's not necessarily the case in the eyes of leadership. So I really am asking this year that if you wouldn't mind taking the time to nominate yourself or someone else to just highlight a member for the month. You know, hey, they joined this club outside of the organization. Hey, they cleaned the worked on this validation, which was super crazy and had so many problems along the way. And of course, I will check with that member if they're okay with being highlighted. But I would love to love on them. So please, for this year, that's just something new that I would love to do. So please take that into consideration. All right, so I'm going to start with Dr. McCarter, who has some announcements about our annual First Coast Infectious Disease Clinical Microbiology Symposium.
And I'll try not to yell for the people on Zoom. I'll project, right? So, hey, everybody, just a reminder that the 31st annual First Coast Symposium, next February 23rd and 24th, at the same venue as this past year, so the Hyatt Regency Jacksonville Riverfront. The program should be out this week, hopefully, if I can get our webmaster to post it. And so we've got a lot of awesome speakers. And what am I forgetting? I was hoping, honestly, to have the website up for registrations tonight so that we could start registering as early as today because people are excited. But I'm hoping within the next week or so, it'll be ready to go. Anybody have any questions or anything? The 23rd and 24th of February. So it doesn't compete with, I don't think, maybe the Super Bowl. And it doesn't compete with Valentine's Day. And I don't know if it competes with Bike Week or not. We've competed with Bike Week before, so that's the only reason I say that. So, okay, cool, thanks.
Thank you. Okay, any announcements from the membership? I don't really want to. Oh, yes, Terry.
Oh, yes, for the Zoom folks. Hi. For those of you that don't know me, I'm Terry Ellis. I work over at the University of North Florida, where I am training many of your aspiring technicians to be. We are having problems in our microbiology lab sourcing media. Media is being discontinued, and the stuff that is left costs an arm and 3 legs. So we are, and we do not have the purchasing power of a hospital to negotiate with. The vendors ignore us. So I am turning to you guys. If you know anybody that you could give us a letter of introduction to a good vendor and sort of kick them in the rear and say, negotiate a decent price with them, or any other ideas on how to source the media that we need for our basic microbiology course for the lab, we would greatly appreciate your help. Thank you.
So just a question, would expired media help in any way?
Very possible.
Okay, the answer was yes. Great. So yeah, any laboratories out there that have expired media, unfortunately, we deal with this problem quite often. Please let myself or Terry Ellis know. Thank you, Terry. Any other announcements from membership? So we have ID rounds every month. I'm not promoting myself, but I happen to be talking tomorrow on blood culture collection reduction, contamination reduction. So if you're interested, let me know. And let's see. So next we will move on to the membership report and finance report, as well as our CEU process shared by Matthew Schminti.
Hello. So far we don't really we have maybe 30 members, I would say, about there. Oh, the end of last year, I believe it was 77. Yeah. And what else? CEUs obviously will be done by me. We want you to be paid by the end of October to stay in good standing, so don't forget that. But that's about it. Not too much going on yet.
Well, how does the CEU process?
Oh yeah, so I'm gonna put September's in pretty quickly, but then going forward, I'll just check them against being in good standing.
So as long as you sign in or you're logged into the Zoom, you will get credit as long as you're a in good standing JAM member. And so we remain at $25. And also just know that if you have anyone who has any interest that may not be able to attend, we will continue Zoom throughout the whole year. So please encourage them, let them know that they can join at any time via Zoom. Yes, ma'am. Thank you for asking that question. Yes, all of our JAM meetings are recorded unless for some reason a speaker has requested not to, but majority of speakers do allow for recording. And that is available. Sometimes there is a little bit of a lag, but those will be available on the jamsociety.net. So correct, but we wouldn't know that you viewed it, so you would have to email Matthew. Yep. Yes. Correct. You need to notify Matthew. Yes, that is a great question. Anyone else have any comments or questions, concerns? Okay, wonderful. So next we will have Beth, who will speak on our, who I'm sure very much loved RSVP process. Beth, where are you? Where did I lose you? Oh.
So we started a new thing this JAM season so that since we're doing both hybrid presentations by Zoom and in person, and we want to let you all feast when you come visit with us in person, we need to have a good idea of how much food to provide. We don't want to waste JAM funds by providing more food than there are people to eat it, and we don't want people to go hungry because we're not getting enough food. So I, in my email body, will ask for our RSVP if you are planning to come in person. There's no need for those of you on Zoom, always, no need to reply if you're not going to eat with us. But in order for us to be responsible with the funds that we get, if you could please RSVP me, I will make a chart. We'll give whoever's in charge of the food the numbers of the people who are going to be eating. And hopefully we will meet without exceeding or not having enough the needs of the meal of each night. So please don't not read your email. Let me know if you're coming.
Thank you, Beth. And of course, if there's a last minute change, don't not come to the JAM meeting. There will probably be extra food because, you know, those estimates are always a little bit larger than they should be. Okay, let's see. I think I have everything. And the most important, Hologic has graciously agreed to sponsor this meeting. So a thank you to Hologic goes out to Shane McCann and his team that are here with us tonight. So thank you, thank you. Dr. Morgan Bartram can answer any sciencey questions, and Kara is open to any, if you have their instrument, question her on the ins and outs on it or any problems you're having, share that with her tonight. So without further ado, Shane said he didn't want to speak, so I'll let him be. And we'll go ahead and have Dr. Hada present our speaker for tonight.
Thank you, everybody. So, hi, Jam. It's good to see everybody this year. I'm looking forward to it. We're back. So it is my pleasure to introduce Dr. Pieper from the Anastasia Mosquito Control District of St. Johns County. He is the molecular biologist and entomologist, received his bachelor's degree from BYU, Idaho in ecology, wildlife, and fisheries. That sounds fantastic. His master's degree from Frostburg State University in wildlife and fisheries biology, and his PhD from Texas Tech in environmental toxicology, where his studies focused on vector-borne diseases, a favorite of many of us in the room. Dr. Pepper then completed a postdoc at Texas Tech, where he focused on the public health consequences of vector-borne diseases. And part of that postdoc, he worked as a BSL-3 lab technician in the Bioterrorism Response Laboratory. Dr. Pepper has 24 peer-reviewed journal articles and 3 book chapters published, and has presented his research over 27 times at regional, national, and international conferences and JAM. Fun fact, he has worked with a variety of animal species over the years, and throughout his research has included bears, river otters, fishers, beavers, bats, quail, deer, and wild pigs. So I think that pretty much covers everything we could ask for. With that, Dr. Pepper.
All right. All righty. All right. Awesome. All right. Well, thank you so much for the invite tonight. I'm real excited to be able to share a little bit about my passion. As you might have guessed from my background, I didn't actually get into this field till probably partway through my PhD. But when I did, I quickly fell in love with it. Arboviruses, vector-borne diseases has quickly become a passion of mine and something I love to talk about and try to bring awareness to. So, as was mentioned, I guess a lot of you have a lot of background in vector-borne diseases, so some of this might be a little bit of a review, but I'm still going to jump into it. So, this data is a little bit old, so I'm sure these numbers are a little higher now, but there are roughly 1,400 known infectious pathogens, over 300 of which are of clinical importance, and about 88 are known to be vector-borne. Annually, I think these stats are mind-boggling. Half of the world's population is at risk. 1 billion people are infected, and over 1 million people die on an annual basis from vector-borne diseases. And they continue to be emerging and re-emerging over the years. So just because you might get rid of a specific vector-borne pathogen, don't hold your breath, because it'll definitely come back. A pretty famous report from the CDC that came out a few years ago reported that from the years of 2004 to 2016, the number of vector-borne disease cases in the United States tripled. Now, if you can look at the top graph there, you can kind of see that spike in 2016. Obviously, that was because of Zika. Yes. So even with that outlier, if you were to remove that data, the number of cases still more than doubled. And during that same time frame, 9 new pathogens were discovered or introduced into the United States that we know of. And that is very alarming. So a vector. So a vector is an organism that's capable of transmitting an infectious pathogen from one host to another. And there are a bunch of different kinds of transmission: animal to animal, human to human, or animal to human. A couple different types of transmission. You have the mechanical. One thing to note with this is that the pathogen, with mechanical transmission, the pathogen never actually enters into the vector's body. Meanwhile, with biological transmission, the pathogen is harbored within the vector's body, and it requires the vector to be able to complete the life cycle of the pathogen. Some characteristics that increase the vectorial capacity of different vectors is, the main thing would be the blood feeding behavior of different vectors. This, as I mentioned, will increase that vectorial capacity as it's able to inject the pathogen directly into the bloodstream for most cases. In some cases, like with Chagas disease, the vector will deposit the pathogen in its frass right outside of the bite, which then typically is like scratched into the wound afterwards. So it still enters into the host body. Again, some different characteristics that increase this vectorial capacity. Vectors that are people loving. It's amazing to see different vectors, even within, say, like a mosquito. Different species of mosquitoes are attracted to different types of hosts, and there are some that specifically seek after humans. So that will increase their vectorial capacity. Some just love animals. And then you have the nasty ones that are opportunistic and bounce back, but they don't care what they feed on. And I feel like those are the worst of the worst. So it's important to know your vector, because different vectors will, especially when, like with a physician who's trying to diagnose a sick patient, right? Knowing what kind of vector they were in contact with can guide their diagnoses. For instance, if a patient knows that they were bit by a tick, that can lead the physician to know it's probably going to be a bacterial infection. However, if they know they were bit by a mosquito, it's most likely going to be a viral infection. Also knowing the time of day. So with mosquitoes, if a patient knows he was bit by a mosquito and he knows it was during the day, that can lead the physician to realize it's probably, though you can't guarantee it, but it's probably most likely Zika, dengue, or chikungunya, and you can rule out some of the other ones. However, if it was during the crepuscular or nocturnal time frame, it's more likely going to be West Nile, St. Louis, or perhaps malaria, depending where you are in the world. So knowing these different factors about vectors is critical to the public health. And then, of course, there's a social economic burden globally with vector-borne diseases. You know, social economically, when an individual is infected with a specific pathogen, it hurts them financially, right? It prevents them from going to work. They accrue medical costs that are crazy. It also hurts businesses and governments as productivity and working days are lost as these patients are stuck at home. And a good example of a global zoonotic disease burden is Chagas disease that we mentioned earlier. It's estimated that annually $188 billion are lost due to this disease. More of a national scale, it's estimated around 41 billion is lost annually in the United States. Contributing towards that would be Lyme disease, roughly $500 million. West Nile virus, there have been several studies with this one, and the different studies average out to roughly $35,000 per case. However, if you look at West Nile fever, it'll probably end up costing a lot less compared to like a West Nile neuroinvasive disease. That'll put a lot harder of a burden on a patient. Now, some basic examples of different vectors. Obviously, you have the ticks, fleas, true bugs, And then the one I deal with every day is the mosquito. And so tonight I'm going to focus a little bit more on the mosquito. So I'll be honest, before I started, this field during my PhD, I couldn't have told you there was more than one species of mosquito. All I knew was I hated mosquitoes, and I thought every single one needed to die. But it turns out there are over 35,000 species of mosquitoes worldwide. Just in North America, there's roughly 175 species. And in Florida, there are roughly 80 to 90 species throughout our state. Now, when you ask someone what the world's deadliest animal is, especially a kid, I think, they would probably respond with something with fangs, something that's fierce. When in reality, did I hear hippo? Hippos do kill a lot, but not as many as the mosquito. This was put out by the Gates Foundation a few years ago. And as you can see here, and this is actually what a lot of people believe to be a conservative estimate. A lot of people believe that it's over a million deaths every year just from mosquitoes. And so mosquitoes alone are known to carry about 38 clinically important pathogens. On an annual basis, it's estimated that about 390 million cases of dengue exist and over 240 million cases of malaria. So in the United States, some of the mosquito-borne cases that we see, the top one is dengue every year. However, a majority of those cases are travel related. So that's an individual traveling to a dengue endemic area. They come back, they get sick, and so they're diagnosed here, but it's reported. Again, next, because of Zika, just the 1 year, that on this graph bumped them, the number of cases that we had in the US and in the territories bumped them into second place with just 1 year of data. Now, the first one that we actually have that is endemic in the United States on this graph, and where we have local transmission everywhere throughout the country, is West Nile virus. So vector-borne diseases are known to be grossly underreported or misdiagnosed. This topic specifically is my main passion in the field, is trying to help physicians understand the importance of trying to get an accurate diagnosis when it comes to vector-borne diseases. So some of the reasons why a lot of vector-borne diseases are misdiagnosed is because they do all kind of present the same with the same symptoms. And so they're often misdiagnosed as, the common cold or the flu. A lot of people will say it's an unexplained summertime fever, or whatnot. So, if someone has cold or flu symptoms during the summer, they'll just call it the summer flu, when in reality, it might be a vector-borne disease. And so an example here, we'll take West Nile virus. So, and so when West Nile virus is diagnosed, it's reported to the CDC. So we have some numbers here. So from 99 to 2017, over 25,000 West Nile fever cases were reported to the CDC. West Nile neuroinvasive disease, almost 23,000 cases. So the CDC also reports that about 80% of those infected are asymptomatic, so you never know. 1 in 5, about 20% will develop the West Nile fever, and a little less than 1% will actually develop the West Nile neuroinvasive disease. So taking those numbers, for every 1 case of West Nile neuroinvasive disease that you see, you would expect there to be roughly 28 cases of West Nile fever. So again, based off of the numbers that were reported to the CDC, you would expect, based off of the WNND cases, that there would be over 657,000 cases that should have been reported to the CDC, but weren't. So that's over 96% of likely West Nile fever cases are not being detected or reported. Now, there is a caveat to that, because obviously everyone who's sick with the cold or flu isn't going to the doctor, and so it wouldn't be reported or tested. However, I would venture to say it's not 96%, that aren't doing that. I feel like a lot of these cases are being misdiagnosed. And so we kind of ran, when I was at Texas Tech, a little pilot study with physicians. And so we sent out a questionnaire to a group of physicians, and we asked, and we laid out kind of different signs and symptoms of a patient and asked them to diagnose them with their top 2 choices of what they think this patient had. And so we pulled together, so like in this instant, we pulled together West Nile fever. And as you can see here, not a single physician correctly identified West Nile fever as their first or second guesstimate for their diagnosis. We did this with several others. We did get, some more positive. We got some correctly identifying it with their first and second diagnosis with some of the other ones. The one that we had the highest success with was dengue fever. A little over 50% correctly identified that on their first case, which I guess is good since that's the #1 disease case that we see in the United States. We also, during that study, gave physicians an opportunity to kind of give some free response and whatnot. And it was striking to see how many physicians actually kind of commented, and they're like, they don't care. Yeah, literally, they said that. If it's viral or bacterial, they treat symptoms. So it's symptomatic and supportive care. Once they realize it's viral or bacterial, they move on to that. And in my mind, biased mind, I guess, I feel like that is the public health ramifications of that are drastic. And I feel like the physicians don't quite understand the importance of getting that accurate diagnosis. So having an early detection of the vector-borne infection can have a potential reduction of the overall incidence of those disease cases. It gives the opportunity for mosquito control, which I work at, an opportunity to get out there and try to knock down the vector population before they have an opportunity to infect more and more people. This is especially worrisome for some vector control jurisdictions. They won't actually go out and treat mosquitoes until after there's been a confirmed human case in their district. Yeah, I don't agree with it, but so if that is their policy, if these human cases aren't being identified and accurately diagnosed in a timely manner, then these mosquito control programs aren't doing what they need to be doing. So let's dive into vector control a little bit. 2 main purposes, I'd say, of vector control. First, to control nuisance mosquitoes. Everyone, I'm sure, has been out. I mean, we're all from Florida. They're awful. They can ruin a picnic quicker than anything. But then also a big part of vector control is to help prevent and control the vector-borne diseases from occurring within their jurisdiction. They do that by controlling the vector populations, monitor and source reduction, population reduction. And this helps to prevent the goal is to help prevent the human disease cases. So as an example, the first really effective control that we saw with mosquito control happened in the early 1900s in Cuba with yellow fever, and also with the development of the Panama Canal with yellow fever and malaria. The Panama Canal was actually shut down. Like they quit partway through construction of that because of the vector-borne disease cases. They were wiping out everybody. People couldn't stay alive long enough to actually complete the construction of the Panama Canal. So it wasn't until the United States stepped in and were able to bring some mosquito control operation, they were then able to complete the Panama Canal. And the efforts of mosquito control were enhanced in the 1940s with the development of synthetic pesticides. In fact, it had such great effect that by the 1960s, a lot of mosquito control programs were getting defunded. A lot of people were like, well, you did your job, but we're good now. We don't need you. They defunded mosquito control. And that turned out to be not such a great thing to do. So there was a recent report by the CDC. They stated that they surveyed a bunch of mosquito control programs, mainly through the South, Southeast, Southwest. And 80% of respondents actually indicated that they are still underfunded and that they lack the required resources to be able to effectively do what they need to do. However, recently, as we saw with Zika virus, I feel like the importance of vector controls is once again being recognized. So you can see here, again, just in 2016, we had over 41,000 cases of Zika. With the aid of mosquito control or vector control entities, in 2017, the number of cases were drastically lower, and then in 2018, almost non-existent. And that's very encouraging to see that mosquito control, though is able to have such an impact on a public health problem that we had. So I want to jump in now into some of the molecular tools that we have in mosquito control. All right, so these different molecular tools, they help aid in the rapid detection of viral activity in an area, which again is very important to a healthcare system as well. I believe that there needs to be a strong line of communication between vector control and public health or the healthcare community, right? So as soon as in our screening and mosquito control screening, if we detect a positive mosquito pool or a positive chicken or something like that, we should be contacting the healthcare and make them aware, like, hey, there's viral activity. Perhaps you can start considering this more during your differential diagnoses with your patients. And likewise, if health or a physician diagnoses a vector-borne disease in a patient, ideally they'd be contacting vector control and be like, hey, you need to get out there and do something with the mosquito population. So I conducted a little study where I surveyed vector controls in Texas and Florida, and it turned out that about 81% of those districts actually said that they reported whenever they found a positive to the healthcare system. 75% indicated that they received notification from the healthcare community. So I feel like both of those should be at 100%. We need to have a strong line of communication between these entities to help fight the public health concerns that we might face. Okay, so one of I would say probably the most popular tool for testing mosquito pools for arboviruses is PCR. Obviously you have traditional PCR. For years when I was in Texas, we just used traditional PCR. I'm not a fan. I don't know what you guys use. I think, and again, this is Steve Pepper's opinion here, but I feel like traditional PCR, it's a lot slower of a process time. There are multiple steps needed. You run your PCR, you run it out on the gel, but I also feel like it's a lot harder to interpret the results. A lot of times you can get a really nice, crisp, clean looking gel. Other times you get some looking like that. And what I think is a positive band, you might not. And so depending who's actually reading that gel, I feel like there's a lot of bias with that. And so I prefer the real-time PCR, a lot quicker results, which again is beneficial to the operational control of mosquito control, right? The sooner we find a positive pool, the sooner we can get the guys out to go spray. I also feel like it's more sensitive and reliable. It's able to capture a lot more false negatives. And an example from that, so I'm pulling a lot of experiences from Texas, but a little study we did there. So when I was working in the lab there, like I said, we used traditional PCR for years. And we kind of did a two-step process. We first screened our mosquito pools for a flavivirus consensus, using a flavivirus consensus primer set. And if we got a positive on that, then we ran it with West Nile virus specific primers. And so over those years, we had 42 instances where we had positive flavivirus, but negative West Nile virus. And towards the end of my time there, I was finally able to convince my PI to get a real-time PCR machine in the lab. And so we went back and checked all of the samples that we had gotten to see a comparison. And so of those 42 that were Flavi positive, West Nile negative with traditional PCR, we were able to detect 9 additional that were actually West Nile positive. And so again, you have the public health consequences of that, is, you know, as in you have that viral activity, but you're missing it because of the molecular techniques that you're using aren't actually able to capture what you need to capture. So currently at AMCD, I run a triplex with my PCR for mosquito pools. I look for West Nile virus, St. Louis encephalitis virus, and Eastern equine encephalitis virus. At first, I actually got a little bit of pushback for the fact that I was screening for triple E. I work in a BSL-2 lab. And with all the pushback I got, I started, you know, asking around and talking to people. I contacted the CDC. Turns out you are allowed to screen for triple E in a BSL-2 lab. You just have to, there's some steps you have to take if you detect a positive sample. So if you detect a positive sample, which I'm sure you guys deal with this a whole lot more than I do, but so you might be more familiar with this. But if you do detect a positive triple E sample, you have to report it to the CDC within 7 days. You have to use their specific form, which is that, and you have to maintain that record in your labs for 3 years. You also have to destroy the sample within 7 days, or if you have an agreement with approved BSL-3 lab beforehand, you can transfer it to them, but there are some challenges with that. But so if your plan is to just destroy the sample within the 7 days, you need to have a destruction protocol in place so that you're not, you know, frantically running around trying to figure out what I do with the sample after you have the positive hit. And then you also have to maintain storage of the sample until destruction. You know, that helps protect against theft, loss, or accidental or purposeful release. So with the mosquito pool testing that I've been doing, so I didn't add any of my data from this year, but so my first 3 years at AMCD, we only had 4 positive pools. They were all West Nile virus. And this was very surprising to me. In Texas, we would have 4 pools from one trap site test positive in a week. Like, we had positive pools all over the place. And I found it very interesting that in talking with other people, apparently in Florida, Panhandle's a little different, but you really don't detect positive mosquito pools very readily. There's some debate as to why that is, perhaps just because the number of mosquitoes are so much higher here. So the chance of you actually finding a positive pool is less. But so, you know, I found that very interesting, and we're still kind of doing some other studies in-house to try to see how we can increase that potential detection. It's not looking good so far, though. There are some other tests that some districts use to screen mosquito pools. Some use what's called a ramp, and it's pretty much you process your mosquito pool, you put it in on that little cartridge, and you pop it in the machine, and it'll, I can't remember, it's like 5-10 minutes, it'll give you a result. Others use the VEC test, which is essentially a dipstick assay. These tests are a lot quicker, but there are several studies out there that show they are not as reliable, especially compared to a PCR assay. However, some districts, you know, especially smaller districts, might not have the capability to have in-house PCR testing for whatever reason. And so, though these tests actually are kind of more expensive than I was expecting, they probably are cheaper than PCR. So some of the districts are forced to go with a less reliable assay than what they probably would like. So the other thing that we really focus on is the Sentinel chicken program. I love the Sentinel chickens. A lot of people don't, but so it turns out that we get a lot of positives with Sentinel chickens. And intuitively, when I got here, I was like, oh, you should be able to find, a positive in mosquito pools way sooner and way faster than you would in a sentinel chicken, right? Because you have the viral activity. It takes a week or so for the chicken to develop the antibodies you're actually screening for. But like I said, the PCR isn't quite working out yet to be that effective. And so we have to rely on these chickens to find viral activity in our county. I think mainly it's like Florida and California are the main states that actually use sentinel chickens for viral detection. There are a few other, there's like Galveston, Texas, I think does it. Delaware might do some, but really it's not that common. But in Florida, it's used a lot. So chickens are what's considered a dead end host. So when a chicken gets infected with West Nile virus, there's no concern of it. It doesn't develop viremia high enough for a mosquito to then pick it up and give it to us. And so it is considered safe for the community. And traditionally, testing in Florida, traditionally in Florida, testing has been done by the state. However, right when I got here in 2020, we had COVID, right? And so all the state labs that were doing sentinel chicken testing, all their resources got shifted, and they weren't doing any sentinel chicken testing for the beginning of that year. So that left a lot of mosquito control programs who relied on that information kind of out of luck. Luckily, we had funds available, and we were able to pursue a method to develop this in-house. And so we were able to, in the middle of 2020, we were able to get our chicken program up and running. So with the help of another mosquito control program, Lee County down south, we were able to identify this commercial kit, a competitive ELISA for West Nile virus. Downside of it does come from France, and so you have to get an import permit with the USDA. You have to file a Toxic Substance Control Act certificate with your shipper, FedEx. So once you get that out of the way, you get your kit. I love this kit. Like it's super easy to run. It's very reliable. And we'll talk about that here in just a minute. In comparison so the testing that the state does, it is a lot more robust than what we're able to do in-house. And so the state initially screens our chicken samples using a hemagglutination inhibition assay. And if you get if you get an equivocal, reactive, or presumptive positive, then they move on and test it using an IgM ELISA. Then if it's equivocal so if it's positive, they report it out as positive with the ELISA. If we get an equivocal or inconclusive, then they'll move on to a PRNT test for confirmation. And so again, it's a lot more robust. However, there are some downsides to it. So the pros and cons of doing the sentinel chicken assay in-house. Cons, obviously there's an associated cost. With the state, pretty much you only had to pay to whatever it costs you to bleed the chicken, and then the $13 to ship it to the state. And then they did the testing for you at cost. Like we didn't have to pay for anything. So moving it in-house, you obviously have to buy the equipment if you don't already have it. So that's a startup cost. But then if you buy in bulk, you can get the assays down to about $3.50 per sample. So that really adds up. Like we have about 54 chickens that we test every week. And so, in the long run over the season, that can really add up for a district. We also are limited to, we really only have a good, reliable assay for West Nile virus. So West Nile and Triple E are our main arboviruses that we deal with in St. Johns County. So we really need to be able to find a Triple E assay that we can use in-house. The pros, as I mentioned, it's a way faster turnaround time. So we get results same day. We bleed the chickens on Monday. We get results Monday evening before I head home. With the state, typically, if everything goes smoothly, we ship it Monday night. It gets there Tuesday, and they report results out on Friday. However, there are sometimes you have unexpected delays that can delay that out to several weeks. And then when you're waiting for a confirmation, that can be even longer, weeks to even months at time. And some districts, when they use the state lab results, a lot of them won't go out and treat the mosquito until they get a confirmed positive chicken from the state. And so by that time, if it really did take a month or so, there's no point. That viral activity has been there. It's been going, and so it's really just lip service at that point, I feel like. And again, so having that quick turnaround in results is very beneficial operationally, right? We get results Monday. We can send the technicians out Tuesday to spray the area if needed. The test is also, I found it to be very reliable, very accurate. And here's an example. So in 2020, we had this chicken that tested positive. Oh, that's small. So it looks like September 8th, we got a first positive hit on our in-house assay. And at this time, we were sending, so the state was back up and running. So we were sending aliquots of our sample to the state for them to test. We were trying to kind of validate our test against theirs since it was new in-house. And so with the state results, they didn't come back with a positive on this bird until the middle of October. So 5 weeks where we continued to test positive in-house, the state was actually reporting it as negative. And then because we were waiting for the confirmation to come on this sample, we actually kind of had some back and forth. The state reported it as positive, then it was negative, then it was positive, then it was negative. So that was very confusing. They eventually were able to confirm this bird as positive. We received that confirmation on December 11th from a sample that was collected November 30th. So that's not even on this graph when that was collected. And so I found that, so that was my first positive bird from being here. I was like, well, this is not good. We had some issues with this. So I took that sample and I was like, maybe we can try to confirm this bird in-house. And so, I took a bunch of, I did RNA extractions on the serum from the week it tested positive and I think 3 weeks previous, just to see if we could get a positive hit with PCR. And luckily we did. We got a positive from the week before, which intuitively makes sense, right? You expect a bird to be infected and then the immune response and the development of the antibodies. So that was very encouraging. So I was like, all right, great. Let's do this with all the birds that test positive moving forward. So in 2021, results weren't as great as I was hoping. We got about 61% of our birds I was able to confirm in-house with PCR, compared to the state who only actually reported about 38%. However, 2022 hit and everything got flipped upside down. Doing the same method, I only was able to confirm in-house about just under 23% of my birds, while the state had almost 73%. So this was very confusing to me. But then I kind of sat down and thought about it. So viremia is often limited to 3 to 5 days in a sentinel chicken. So if we're only drawing blood every 7 days, obviously there's that chance that we could be missing the actual viremia, and the samples that we're collecting, we won't be able to get a positive through our PCR. So potentially that could explain some of the issues we had with actually getting in-house positives. I've actually been talking with the state, and they're kind of looking into this as well. And so we're trying to find out how we can improve using PCR as a way to confirm these birds, whether or not it'll be a reliable process moving forward, as it'll definitely help speed things up. Because again, if I get a antibody positive chicken on Monday, Tuesday I can come into the lab, do an extraction from the previous week's serum sample, and I can get it confirmed or not, on that day. So 2 days we got a bird positive and confirmed, instead of having to wait, the inevitable. I'm not trying to throw the state on the bus. Obviously there are a lot of issues with, you have to, which I'm sure you guys deal with, you have to ship your sample off, and then labs have a lot of samples to test. And so there's an inevitable delay that you will experience with that. So despite being limited to only West Nile virus, I do feel like doing in-house testing is very beneficial to the operational control of mosquito control. You know, I still encourage everyone to send samples to the state, because they also they are able to test for more pathogens than you are at home. So that's beneficial as well, especially when it comes to triple E. So even though triple E is rare, if you get it, it's bad. So we want to try to do the best we can to be able to control triple E. And ideally, I would like to try to be able to develop, as I mentioned earlier, an in-house triple E ELISA to be able to use. So now to close out here, I just wanted to spend a little time kind of going over malaria. I know malaria has been a kind of a hot topic right now, especially in mosquito control. And so let's dive into this a little bit. All right. So malaria is considered one of the most severe public health problems worldwide. In most reliable data I saw was in 2020, over, as you know, we mentioned earlier, over 240 million cases were reported, with over 627,000 deaths occurring. That's awful. And a lot of these cases, they mainly affect children and pregnant women. And so it's just, when you hear malaria, it's just, it's gut-wrenching, I feel like. So there are actually over 100 species of Plasmodium around the world. However, only 5 of those are known to infect humans. The vector for malaria is the Anopheles mosquito. There are over 430 species of Anopheles. However, only about 30 to 40 actually play a role in malarial transmission. So again, it's kind of why it's important to know the different mosquitoes and species that are in your area that can help determine whether or not, you know, malaria might be a cause. So despite the early history of malaria in the United States, nowadays it's typically not considered a major concern in the US. It was actually considered eliminated from the United States in 1951. So going back to this, the table we had up earlier, just after West Nile virus are actually the number of malaria cases that we experience in the United States every year. So roughly 1,500 to 2,000 cases we get every year. However, majority of those, just like dengue, are travel related. So having a quick and accurate diagnosis of malaria, or honestly any vector-borne disease or any disease, is very important. So because it's able to help prevent further infection within the population. You know, the earlier we're able to get an accurate diagnosis, the earlier we're able to break or even potentially prevent local transmission cycles from occurring. So delays in diagnosis and treatment is actually the leading cause of malarial deaths. So the sooner we're able to get an accurate diagnosis, you're able to treat sooner, and malaria is treatable. And so if there's a delay in that, that's when we actually see deaths occurring with malaria. So some diagnostic testing for humans. You got the blood smear microscopy, which I believe is considered the gold standard for malaria testing. I don't do it, so I could be wrong on that. Correct me if I'm wrong. But then there are also rapid diagnostic tests available. So obviously in the name, they're rapid. We're able to get a quick result. However, if you get a positive or if it comes back negative, you still have to get it confirmed with the blood smear. So it can kind of perhaps guide your diagnostics, but you can't just fully rely on those rapid tests. And then you also have the PCR testing, which again is more sensitive, but again, it takes longer to get those results. However, the PCR testing is very beneficial because you're able to confirm the exact species of malaria that an individual is infected with. And this helps a physician to be able to determine which treatment options they need to use with that patient. So diagnostic testing with mosquitoes. I'm pretty sure the main one that's used is again PCR. I've been trying to get a hold of protocols to kind of have in my wheelhouse in case any different arboviruses or pathogens come in our district, I want to be able to be set, ready to go, so we can test our mosquito populations. I've been struggling to find a good protocol for malaria. I do feel like it's mainly, all the testing is mainly done at the state or more so the federal level. And so jumping into 2023, we've all heard, about the malaria cases, the local transmission of not the travel related, but the local transmission that we had here in Florida. So I believe this is up to date, where 7 cases were confirmed to be locally transmitted in Sarasota County, Florida. So these individuals, they had no travel that would have indicated that they would have picked it up from somewhere else. And this focal transmission zone was about 5 miles squared in Sarasota County. So it's not like the whole county was, infected with malaria. They were able to identify the infection as Plasmodium vivax. So this is a less virulent strain of malaria. However, it is the most widely distributed. And even though it's less virulent, again, if there is a delay in getting the diagnosis, it still can cause severe disease or even death if it's untreated. With these cases, they were also able to detect that it was Anopheles crucians species that were the vectors for the malaria cases, and that testing was done at the CDC, I believe. So this is why vector control is so critical. So knowing what vector species are present in your community can help identify whether or not, so like if you have a patient come back with malaria, and if you have that vector species in your community, you can very quickly become established, and you can get that local transmission. However, if you know, like, hey, we don't actually have the vector that would vector malaria in our area, then it's not such a drastic deal. Obviously, it still is to the patient, but you don't have to worry as much about there being that local the threat of that local transmission in your community. It's also being able to advise public health agencies on potential any advisories that they send out regarding cases like this, like malaria. For instance, knowing that with malaria, it's the Anopheles vectors, they bite at night. So in these advisories, the health departments can include in them, like, hey, if you go out at night, that's when you need to make sure that you're protected. Wear the long sleeves, wear the DEET, or just stay indoors if possible. Or if it's like a dengue case or something like that, where during the day, we're able to help the public health entities specify the specific things that people need to look out for in these advisories. And again, as we mentioned before, able to alert medical professionals of active viral activity. And again, hopefully this is a back and forth. If we detect it, we notify you. If you detect it, you notify us. Because again, the end goal is to limit or prevent as many vector-borne disease cases as positive in our community. And with that, do you have any questions? If we have time.
Thank you, Dr. Pepper. Any questions from the audience, whether in person or online? I'm sure y'all have questions. Yes, Dr. Hada.
This is great. I really appreciate hearing all this information. In terms of vector control, so if you have, say, a positive mosquito control, a positive mosquito pool, or a positive hit on a chicken, how do you know where to start spraying or do your other efforts? And how big of a zone do you do?
So it's dependent. So it depends what your positive is for. So again, depending on what pathogen is being vectored, different species of mosquitoes will fly or travel different distances. So some, like with the the 80 species that vector dengue or Zika, they have a very small range. And so a lot of times vector control would go and just kind of spray around an individual's house. If it's West Nile virus, those mosquitoes can travel further. And so, we spray in a further radius. As far as where to spray, that kind of depends on, so we have different coop sites set up throughout the county. And so we're able to, when we get a positive, we can see where that chicken is throughout the county, where the population is, where the concern is, and where the potential vector mosquitoes will be. And we try to attack it that way.
Do you vary your zone based on the pathogen? And that, do you spray more if it's triple E versus dengue? Does that have a factor?
No, perhaps the way we go at it might change a little bit based on, again, kind of based off of the mosquito that's of concern for the vector. We might do more larviciding versus adulticiding based off of the mosquito, but you don't want to just go out there and, like, if it triple E, potentially more concerning. You don't want to just over blast it with chemical, because that can lead to long-term effects of insecticide resistance, which we're having a huge problem with globally in mosquito control. And so you still have to be very responsible in how you try to treat the positive areas that you get.
Great stuff. Thank you.
Yeah.
Thank you. I live about 2 miles south of the area in Sarasota County.
Oh,
So they've been nuking us for a couple of months. So my question, similar to what Dr. Hatta, your answer to her, are there different variations of larvicide and dullicide, like antibiotics, in terms of resistance, or do you have extra tools in the toolbox?
yes, there are different active ingredients, different pesticides that are used. And I'll be honest, I'm a molecular guy, so I'm not as involved with, like, going out and doing the spraying. And so I don't know all the chemicals as well as I probably should. But I do know that, yes, there are different chemicals, some that are a lot more potent, some you have to be more concerned about depending on what's around you, because of, like, certain areas, we're not allowed to spray certain chemicals just because of, like, if it's what is that called? A preserve or something like that, we have to be careful of. Yeah.
For the issue with the mosquito pooling, have you looked at digital PCR, and has the state talked to you about looking at digital PCR for some of these assays?
No, well, yes and no. Actually, I think when Dr. Qualls came and presented last year, I think she came back and mentioned digital PCR. We haven't had time to, like, dive into that yet. We have a collaboration going with UF down at Vero Beach at looking at some different PCR techniques. I think it just got funded, so we'll be starting that up soon. But so no, I haven't looked into it, but yes, it is something that I want to look into. I haven't done anything with digital PCR.
Any other questions from the audience or from our Zoom participants? Okay, well, we did not intentionally plan for you to come to the state lab, but here we are. So thank you very much for presenting. Also, I want to highlight about the Anastasia. Now, correct me if I'm wrong. The center has the official name is Anastasia?
I'll be honest, I don't know what the official name is. I feel like it changes. It's something like the vector education.
So
stuff,
last year, Dr. Quells shared about how it would be open to the community for museum and for families to come and visit. Dr. Pepper did share that if there's still a group who's interested to visit, they can, just to learn more about the mosquito control program. However, there's still probably another year of work or so for
Yeah.
it to be open to the public, but that is still to come and be excited for. So just wanted to share that. And thank you very much for sharing your
Yeah, sure thing.
knowledge on this. Thank you very much. And I will conclude. Oh, yes. Yes.
Oh, thank you. Thank you.
So thank you to everyone for joining us tonight. Our next meeting will be October 12th at Baptist Downtown. And the topic will be detecting and preventing infections with antimicrobial resistant organisms, presented by Dr. Marie Claire Rowlinson. She couldn't be here tonight, but she is back, and we are happy to welcome her back. So for more information on the rest of the schedule, please visit the JAM website. I did send some updates to Matthew today, so they'll be up there shortly. Please don't forget to send in your RSVPs that you're coming for food, and just please come in general to have a good time. Don't forget the member highlights if you can share those with us as well. Have a wonderful evening. Thank you, everyone. Oh, yes, sign in, please, for your CEUs if you have not. And yes, fill out those evaluations, and for those on Zoom online as well.